Hello and welcome. Let's look at chromatin structure from biochemistry. So, we know that every cell in our body has the same DNA, but not all genes in our body are expressed. So, we need to understand chromatin structure. DNA exists in a condensed chromatin form so that it fits into the nucleus. Now, the DNA loops twice around the histone octamer to form nucleosome, which gives us this bead on the string appearance. Then we have the histone H1, which binds to the nucleosome and to the linker DNA and stabilize the chromatin fiber so that it is condensed and it fits into the nucleus. DNA has a negative charge from the phosphate group, and histones are large, they have a positive charge from lysine and arginine. So L, lysine, arginine, that's the mnemonic. DNA and histones are synthesized in the S phase. Mitochondria have their own DNA. It is different from the nuclear DNA. It is circular and it does not have histones. We also know that mitochondrial DNA is inherited from the mother only, and mitochondrial DNA codes for some oxidative phosphorylation enzymes, which is why if you have problems with mitochondrial DNA, you will have problems with ATP. So think about cells that use a lot of energy, like your eyes, muscles, and brains. So we have clinical examples like Leber hereditary optic neuropathy, where there is sudden blindness in young adults. Then we have MELAS, which is mitochondrial encephalopathy, lactic acidosis, and stroke like episodes. If the problem is in mitochondrial DNA, which is distinct from nuclear DNA, it is circular, it does not have histones, and it is entirely inherited from the mother. Now, one other important thing to understand genetic expression is heterochromatin. So heterochromatin is condensed and it appears darker on electron microscopy. So you can see it here, and nucleosome also. It is sterically inaccessible, thus transcriptionally inactive. If you increase methylation or you decrease acetylation, you can make the genetic material more condensed, hence more inactive. Increase methylation or decrease acetylation will make it more condensed and transcriptionally inactive. So heterochromatin HC, highly condensed. Hidden chromatin. One example is Barr bodies. Women have an XX chromosome. So the inactive X chromosome, Barr body, will be visible in the periphery of the nucleus. Next is euchromatin. Euchromatin is less condensed and it appears lighter on electron microscopy. So here, okay, it is transcriptionally active, sterically accessible. Truly transcribed. Euchromatin is expressed E mnemonic. Now we have changes that we can do to the genetic expression without changing its sequence. One example, DNA methylation. Reversibly changes the expression of DNA segment without changing its sequence. That sounds like epigenetics, and it is. So, DNA methylation is involved in aging, carcinogenesis, epigenetics, genomic imprinting, transposable element repression, X chromosome inactivation, lionization. DNA is methylated in imprinting. Okay, imprinting disorders, Prader-Willi, Angelman syndrome. Now, methylation within the gene promoter CpG islands is going to silence, repress gene transcription. So, CpG methylation makes DNA mute, mnemonic. Now, if you have a dysregulated DNA methylation, then you can get fragile X syndrome, dysregulated DNA methylation, fragile X syndrome. Next, we have histone methylation. So, histone methylation usually causes reversible transcriptional suppression, but it can also cause activation depending on the location of the methyl group. So, mnemonic is histone methylation mostly makes DNA mute. Now, lysine and arginine residues of histones can be methylated. Also, remember, lysine and arginine are the ones that give positive charge to histone. Okay, now we have histone acetylation. So if you remove the positive charge of histone, then the DNA is more relaxed, which means it is more transcriptionally active, it is more open. So you can express the gene, you can make protein. So increase transcription. Now, thyroid hormone synthesis is altered by acetylation of thyroid hormone receptors. So histone acetylation makes DNA active. A acetylation, A active. Finally, we have histone deacetylation. So that is removal of the acetyl group. If you remove that acetyl group, DNA becomes more tight, more tightened, more wrapped closely. So you have decreased transcription. Histone deacetylation may be responsible for altered gene expression in Huntington's. So histone deacetylation deactivates DNA. Deacetylation deactivates DNA. So remember, DNA methylation, if it is dysregulated DNA methylation, you can have fragile X syndrome. If we talk about histone acetylation, then thyroid hormone synthesis is altered by acetylation of thyroid hormone receptors, and histone deacetylation is implicated in Huntington's. Okay, histone deacetylation may be responsible for the altered expression in Huntington's. Thank you.